tornar a Mallorca sempre és especial i és algo guapo, no? I emotiu per jo. Vos faré la presentació en anglès, crec que no seria capaç de presentar la meva feina en un altre idioma que fos anglès. Així que ara canviaré. Today I'm going to talk about the functions of N6-methyladenosine in stem and cancer cells. So the central dogma of molecular biology stated that the DNA is transcribed to RNA and RNA will be translated to protein. However, now we know that this cartoon is quite simple, that there are additional steps, and moreover, we also know that the majority of the RNA will never be translated into protein, what is known as non-coding RNAs, right? And some of these steps are also reversible. In addition, beyond the DNA sequence, gene expression is regulated by reversible histone modifications and DNA methylation, known as the epigenome. And beyond the RNA sequence, dynamic chemical modifications of RNA, which is imposed by writers and erased by these erasers, recognized by readers, regulate the RNA fate. Indeed, there are more than 170 RNA modifications that exist in the three domains of life and in all RNA species. In my lab, we are interested in a study N6-methyladenosine, or methylation at the position 6 of the adenosine, which is the most abundant internal modification on mRNA. These modifications is deposited co-transcriptionally by METTL3, which has the enzymatic activity, and to do so, it needs to bind to METTL14. METTL14 is important for the formation of the core methyltransferase. I think there is un micro que no està en mute i sent algú xerrar. No sé, bueno, continuaré. The METTL14 is required for the formation of the physical structure of the catalytic center. So METTL3 cannot methylate mRNA without the binding of METTL14. In addition of this core complex, METTL3 and METTL14, there are other writer proteins that are important for the specificity of the mRNA that are going to be methylated. This chemical modification, M6A, is reversible. That means that it's erased or is demethylated by FTO and ALSBA5. And the fact that it's reversible means that it's subject to regulation and also is a very interesting pharmacological target. M6A on mRNA will be recognized by different readers. And depending on the reader, the fate of the mRNA will be different. It can affect splicing, alternative splicing, export stability, and so on. So this is just this is to illustrate again what I mentioned before about this need of METTL3 to bind METTL14 for the methylation activity. Although M6A was first discovered in the 70s, it has not been until the last decade where the field of epitranscriptomics, which is the field that studies RNA modification, explodes. And why is that? It's because in 2008, Rupert Frey discovered or described the function of METTL3 in plants. A function METTL3 was required for the development of plants. And later on, the same lab developed M6A specific antibodies that help later on to set up this technique, EMI-RIPSIC, by two independent labs, Sammy Jeffrey and Gideon Chavi. So with the EMI-RIPSIC, what researchers had the possibility to map these M6A methylated mRNAs genome-wide. So basically, in this technique, we fragment the RNAs in small pieces, and this fragmented mRNA will be immunoprecipitated with M6A antibodies and subject to sequencing. Very, very similar procedure as in a chip, but with RNA. what we learned from these studies is that M6A is methylated at the consensus motif, a consensus site, and that the distribution of M6A along the mRNA is not random, but enriched at the 3' UTR near the stop codon. So basically, after sequencing, we get an analysis very similar to this. So we have these peaks, and these peaks means here that there is an M6A site. Given that that M6A influence virtually all aspects of mRNA metabolism, it is normal to think that M6A is involved in many biological processes such as adipogenesis, spermatogenesis, development, stem cells, and X chromosome inactivation, and so on. It is also we can also think that aberrant deposition of this mark can lead to many diseases, including cancer. So during the first part of my talk, I would like to focus on the role of M6A in pluripotency and differentiation. For this, I will briefly introduce what are stem cells. Stem cells are derived from the inner cell mass of the developing blastocysts, and has the capacity, these cells, when we culture it in the lab, in IdISBa, they have the capacity to proliferate, to indefinitely proliferate in a property that is called self-renewal. And this is true thanks to the action of many transcription factors, but being the core pluripotency factors OCT4, NANOC, and SOX2. Those cells also, when we have it in IdISBa and we add a special cocktail, differentiation cocktail, those cells also have the capacity to differentiate into the 3 gene layers, endoderm, mesoderm, and ectoderm. So for the differentiation, there is 2 basic requirements, like is to decrease, to shut off the expression of the pluripotency factors, and to activate the expression of lineage markers. So back in 2014, Pedro Batista published a cell stem cell paper showing that M6A is present in many mRNAs from pluripotency factors, not only the core factors like nano.
Ja, pues aquí fer visual i jo que faig imatge.
Quan dona accés amb el micro encès. L'apagam, per favor.
Sí.
Esto se va también.
Per papà.
Perdón.
Vale.
Crec que Gwendolyn encara tens el micro encès.
Sí, voy.
Vale. Vale, venga. Here we can see that important transcription factors such as NANOC, KL4, MYC are decorated with M6A. Notice that OCT4, even if it's a core pluripotency factor, is not M6A methylated, because in green, in gray, this is the signal we would get from an input, right? And the signal from the input, meaning without antibodies, is higher than after immunoprecipitation with M6A. But nevertheless, in red, we have a lot of important mRNAs for self-renewal that are decorated with this modification. In order to study the function of this modification, what Pedro Batista did is to generate knockouts of METTL3. No, sorry. Before that, what is the meaning of M6A on these pluripotency transcripts is that these transcripts are methylated. They will be recognized by this specific reader, YTHDF2, and marked for degradation. Okay, to study the function of METTL3 in stem cells, he generated knockouts of METTL3. And what he saw is that without METTL3, so without the M6A site on these pluripotency transcripts, there was no degradation. These transcripts were highly stable, and those cells lost the capacity to differentiate. they were called hyper-naive, right? High abundance of pluripotency transcript, even if you add the right cocktails, they cannot differentiate. So METTL3 would not affect self-renewal, but affects the differentiation potential of those cells. So during my postdoc, I was interested in study how oncogenes could affect pluripotency, because cancer and stem cells share many of the transcriptional programs that sustain proliferation. So I specifically work on an oncogene called ZNF217 or zinc-finger protein 217, depending if it's a mouse or human species. And what ZNF217 was shown to be is an oncogene that represses the transcription of tumor suppressor genes and activates the expression of oncogenes, at least in breast cancer, and this is true for all other cancers as closely. So what we asked was if zinc-finger protein 217 play a role in ES cells renewal. So for this, I generated knockouts. I was doing knockdowns of zinc-finger protein 217. And what I observed is a decrease in alkaline phosphatase staining, which is a marker of pluripotency. Those cells also show a defect in proliferation, a phenotype in the cell cycle, and increased apoptosis. To better understand the mechanism, we did a lot of cheats because ZNF217 is a transcription factor, and we saw that together with LSD1 and OC4, it was binding to the promoters and enhancers pluripotency factors. But to get like an alternative mechanism, or to gain into the knowledge of the mechanism, we also perform a mass spectrometry analysis of endogenous zinc-finger protein 217. We retrieve already known and published interactors of zinc-finger protein 217, but we also retrieve METTL3. We validated the interaction between zinc-finger protein 217 and METTL3, and as you can see here, when we immunoprecipitate with 217, we retrieve METTL3, and when we immunoprecipitate with METTL3, we also retrieve zinc-finger 217. However, after immunoprecipitation of zinc-finger protein 217, we could not observe METTL14, suggesting that METTL3 could be part of two independent protein complexes, one with 217 and the other with METTL14. As you remember, I showed you this cartoon before. METTL3 needs to bind to METTL14 in order to exert its methyltransferase activity. So this complex METTL3 zinc-finger protein 217 would be an inactive complex. And indeed, in the knockdowns of zinc-finger protein 217, we observed an increase on M6A levels. This is assessed by dot plot, but also by mass spectrometry analysis. And when we did MERIP, we also observed an increase in the number of peaks after knocking down zinc-finger 217. We retrieved the consensus motif, and this increase in M6A sites upon depletion of zinc-finger 217, we call them zinc-finger 217 dependent peaks. They were hypermethylated in knockdowns of 217 and hypomethylated in the knockouts of METTL3, suggesting that both proteins have an antagonist function. Indeed, sorry, this is a bit blurry, but we validated by M6A immunoprecipitation coupled to qPCR, we validate that indeed there is an increase of M6A modification on those pluripotency transcripts after depletion of zinc-finger protein 217. And what happened to those transcripts that were hypermethylated in the knockdowns, they were much more unstable. So they were marked for degradation. So we have a model. I did not show you the top part, zinc-finger protein 217 as a transcription factor, but I show you this lower part in which in stem cells, zinc-finger protein 217 binds to METTL3 and restricts the M6A methyltransferase activity. and thereby it protects those transcripts for degradation. During cell differentiation, when the levels of zinc finger protein 217 decrease, METTL3 combine to METTL14 and methylate the remaining transcripts, pluripotency transcripts that still exist, and this is a requirement in order to exit the pluripotent state and be able to differentiate. Indeed, later studies show that zinc finger protein 217 regulates M6A deposition in other models, such as breast cancer stem cells or adipogenesis. And it has been also widely shown that M6A regulates differentiation, hematopoietic stem cell specification, myeloid differentiation, neurogenesis, and so on. So the summary of this first part would be that METTL3 is not essential for ESL cell renewal, but required for the proper differentiation in the three lineages. And that zinc finger, sorry, this is 217, restricts METTL3 M6A methyltransferase activity. Now I would like to show you some preliminary data that we have from my lab on the role of M6A mRNA modification in cancer. And indeed, not only in cancer, but also in neurodegenerative disease and other disease, aberrant M6A deposition has been shown to lead or to cause several other diseases. In my lab, we focus on breast and ovarian cancer, although we also have a very nice collaboration with Holund here, a colleague working on pancreatic cancer organoids. Ovarian cancer is a collective term for invasive cancers that are derived from different tissues. So it is a very heterogeneous disease, being the high-grade serous ovarian cancer, the one with the poorest prognosis, usually because when it is detected, there is metastasis a bit everywhere. So as you can see here in the cartoon, in the upper side, this is a graphical representation of METTL3 expression levels in different types of cancer. So it seems that there is an increase of METTL3 in ovarian cancer. And then what we did is we took tissues from different of the subtypes of ovarian cancer, and we stained with METTL3 antibodies, and we observed that there was an increase also at the protein level, increased expression in the high-grade serous cancer. To study the function of METTL3, we generated knockdowns of METTL3. We used two differential harpins. And we observed that those cells had an effect in proliferation and increased apoptosis. We then did colony formation assays, and expectedly, also those cells knockdown of METTL3 had an effect in the colony formation assessed by crystal violet. We also perform trans-well migration assays, and in which we have two wells. We see the cells in the upper well. In the lower well, there is a chemioattractant, right? And those cells, we measure the capacity of those cells to migrate in the lower part of the well. And upon loss of METTL3, these cells have lost the migration capacity. Very similarly, we also assess the phenotype after depletion of METTL14, and we also observe effects in proliferation, increased apoptosis, and no changes in the cell cycle. METTL3 is required for migration, as again there is a loss of the migration capacity when METTL3 is knocked down, and also a decrease in the number of colony formed after silencing of METTL14. This is a reminder of the importance of METTL14 for METTL3 in order to form the catalytic center. And what we did is we assessed the expression of the writer complex in the knockdowns here on the left side of METTL3 and here in the right side of METTL14. And what we observe is like when you knock down METTL3 or METTL14, the rest of the complex goes down. And if we focus on this core complex, METTL3 and METTL14, what we can see is that the decrease in the protein levels do not correspond to decrease in the mRNA levels. Therefore, both proteins are required for the stabilization one to another, right? The stabilization of the writer complex. The levels of the other writers, WTAP and VIRMA, does not change either. I did not show for simplicity. What is curious is that HACI, which is a ubiquitin ligase, the levels of HACI do not change when we knock down METTL3 or METTL14. So we have performed cyclohexamide experiments in which we block the translation, and we can observe here, this is the quantification of the Western, that knocking down of METTL3 decreases the stability of METTL14. Then the ubiquitinated proteins, as you know, are usually target for degradation by the 26X S proteasome, which is the major proteolytic machinery for ubiquitinated proteins in the cell. So to assess whether this increase of the stability or decrease of the stability depends on the proteasome, what we did is to treat the cells with MG132, which is an inhibitor of the proteasome. And as you can see here in the knockdowns of METTL3, we observe an increase on the levels of METTL14. So by now we are working on understanding the mechanism by which one protects to the other from ubiquitin proteasome degradation. And to us, well, we are first assessing whether indeed HACI, one of the writer proteins in the complex, would be this ubiquitin ligase. Another cancer model that we study in my lab is breast cancer, which is the most prevalent of the female malignancies, and it is the second leading cause of cancer death. It is a very heterogeneous disease, and in the clinic, the different histological subtypes are catalogized, determined by the presence or not of hormonal receptors. So we have the luminal A that have the presence of estrogen and progesterone receptors, similar as the luminal B, but the difference between these subtypes is the presence of HER2 or Ki67, which is a proliferation marker. Then there is the HER2 positive breast cancer subtype, which of course is positive for HER2 and negative for ER and PR, and the triple negative that, as the name says, is negative for all these hormonal receptors. So the frequency, the treatment, and the prognosis of these breast cancer subtypes is quite different. So we have, this is a representation of the overall survival, and as you can see here, the luminal A is the breast cancer subtype with a better survival, whereas the basal or triple negative is the subtype with a worse prognosis. So the role of M6A in breast cancer has been quite good described. We have all the possible scenarios in which the position of M6A in oncogenes can enhance the breast cancer phenotype or can diminish the breast cancer phenotype. Also, some other works have involved microRNA biogenesis, in drug resistance, but also has been involved in breast cancer, stem cell phenotype of the breast cancer. So we want to understand a bit better what is going on breast cancer. So we are, to do so, what we are doing is to use MCF10 as a normal mammary cell line, MCF7 as a luminal A, which is the one with a better prognosis, and the MDA-MB231 as a triple negative for basal, which is the cell line with which is the breast cancer subtype that has worse prognosis. So we performed knockdowns of METTL3, and we observed that there was a decrease in the cell proliferation, increase in apoptosis, and a decrease in the colony formation after silencing of METTL3. We could also observe that the phenotype was more pronounced in the breast cancer cell lines compared to the normal mammary cell line. In vivo, METTL3 promotes tumor growth. We injected, in collaboration with Ruth Rodriguez at the IdISBa, we injected scramble and knockdowns of METTL3 in the mammary fat pad of nude mice and assessed the tumor volume. And as you can see here in the representation, knockdown of METTL3 leads to a decrease in the tumor volume at day 21. This decrease is correlated with a decrease in Ki67, a proliferation marker, and a decrease in METTL3 expression. METTL3 also promotes cell migration in vitro. So here is a scratch or one healing assay in which we scratch the cells, and in the scrambled cells, after 24 hours, these cells invade this scratching area again, whereas in the knockdown, they have lost this capacity. And in vivo, the same. We engineered a luciferase cell line, and we injected in the tail of nude mice, scramble and knockdowns METTL3 cell lines. And these cells from the tail, they will metastasize in the lungs. And as you can see here, the knockdown of METTL3, they have lost, and this is the quantification, they have lost the luciferase signal. And you can see here the isolated lungs. Again, depletion of METTL3 affects the breast cancer transcriptome, and more differentially expressed genes in MCF7 and in MDA compared to the normal mammary MCF10A cell line. And which were those genes? So genes that were upregulated in breast cancer cell lines were genes related to response to drugs, sulfation in the case of MCF7, and tumor suppressor in the case of MDA, and downregulated genes were corresponding to cell signaling cascades that are important for sustaining the breast cancer phenotype. We perform MERIP assays in which we monoprecipitated M6A mRNA we monoprecipitated M6A mRNAs in the 3 cell lines. And it was very puzzling because what we observed is like, well, there is a core of transcripts that is M6A methylated in the 3 cell lines, and we are now trying to understand what are those transcripts and whether methylation on those sites gives an advantage to the cell lines to become more tumorigenic. But however, we, in collaboration with Eric Dassi at University of Trento, we are planning to do CLIP in order to understand how these sites are recognized by the readers, because at the end, it is not important if an mRNA is methylated or not. What is important to understand is what is the fate of this methylated mRNA, so how this mRNA will be recognized by the different readers. And as you can see here in the lower panel, indeed the readers of M6A, there are more readers, but the most common readers of M6A are differentially expressed in the different cell lines, and this could probably alter the metabolism or the fate of these mRNAs. So the summary of this part 2 is that METTL3 is overexpressed and M6A levels decrease in ovarian and breast cancer cell lines. METTL3 promotes cell growth and invasion and sustain the breast cancer phenotype probably through the deposition of M6A in oncogenes. Sorry. And now I would like to talk about what we think is a non-canonical function of METTL3 in breast cancer. So in 2006, Gregory Lab discovered that METTL3 was translocated in the cytoplasm of lung cancer cells, where in the cytoplasm had an activity that was independent of the M6A methyltransferase. So it could act as a reader of M6A, so recognizing M6A mark mRNAs and allowing the recircularization of these mRNAs, so therefore the ribosomes could be recycled and there would be a much more efficient translation. So this was occurring in oncogenes. So METTL3 in the cytoplasm was increasing the translation efficiency of oncogenes in lung cancer. Diego did a very nice analysis where we were a bit puzzled, right? Because we could not see overexpression of METTL3 in any of the different breast cancer subtypes. But then we analyzed the expression of METTL3 in different subtypes, in different samples from tumors, and what we observed is that indeed METTL3 in breast cancer was also mislocalized at the cytoplasm. So in order to study the cytoplasmic function of METTL3, we fraction breast cancer cell lines, and we immunoprecipitated endogenous METTL3, cytoplasmic endogenous METTL3, and subject those samples for mass spectrometry analysis. We could retrieve other members of the writer complex, such as METTL14, WTAP, and VIRMA, but we could also retrieve this in blue, the exocyst complex, or many components of the exocyst. So what is the exocyst? The exocyst is a protein complex of 8 subunits that are conserved from yeast to human and helps in the tethering of secretory vesicles to the plasma membrane. So which vesicles is that? We are talking about these vesicles that are formed here in the endoplasmic reticulum through Golgi, and then they will fuse to the plasma membrane, delivering proteins to extracellular matrix or proteins that are here will be incorporated in the plasma membrane. We validated the interaction of METTL3 with exoc7 and exoc8 because they were the most enriched proteins in our analysis, and we have a very similar scenario where we immunoprecipitate with METTL3. We detect exoc7, exoc8, of course METTL14, and we did not detect METTL14 when we were immunoprecipitating with exoc7. In collaboration with Marta Valli, we performed turf microscopy, and we could see colocalization of exoc7 with METTL3 at the membrane, at the surface of the cells. In order to understand whether this interaction was direct, we perform GST pull-down assays. And as you can see here, METTL3 physically interacts with both exoc7 and exoc8. So why is this important in cancer? Is because one of the cargos of these vesicles are metalloproteinases that will be secreted here in the extracellular matrix and will degrade. Actually, they will degrade the extracellular matrix, and this is very important to allow the cell to migrate. So this is important in metastasis. And indeed, when we have a cell line, a GFP exoc7, so we can induce, when we overexpress exoc7, the cells really change the morphology. They form these protrusions that resemble neurons, but they are not neurons. They are called lamedipodia, and allow the cell to migrate. So we perform zymographies. I would like you to focus only on the right panel, and where we collect the supernatant in scramble and in the knockdowns of METTL3. And what we observe is a decrease in the activity of metalloproteinases in the media. So there is less secretion of metalloproteinases in the media. We also perform an invadopodia assay in which we seeded the cells on GFP gelatin. And what we are measuring here is the capacity of those cells to degrade the gelatin. So in the scramble, there are these black areas. That means that the cells have been migrating and have degraded the gelatin, whereas the knockdowns of METTL3 have lost this migration capacity. And so here is a inducible exoc7 cell line, in which addition of exoc7 forms this lamedipodia. However, when we knock down METTL3, there is a decrease in the lamedipodia structure, suggesting that METTL3 mediates the exocyst function. So the summary of this part is that METTL3 is overexpressed in the cytoplasm of breast cancer cells, interacts with the exocyst complex, and mediates its metastatic function. However, we are now trying to understand whether this function is totally independent of the canonical function of METTL3 as a methyltransferase enzyme, because usually, both functions are connected, right? And protein shuttle from the nuclei to the cytoplasm. This is a dynamic process, so we are not sure if we are going to really dissect both parts as totally independent items. In any case, I hope I have convinced you that the central dogma is beautifully decorated with chemical modifications, making this cartoon much more complicated, probably much more beautiful also. And with that, I would really like to thank all our collaborators that have helped us in this and other projects. And of course, my lab. This is a work on the breast cancer. It's a hard work of Sirin, a PhD student, and Debbie, a postdoc in my lab. Kanchan here is following up on Ghazala's work on the ovarian cancer. And this also could not be possible with all the funding sources. And I will be very happy to take questions. Thank you very much. Ya está.
Moltes gràcies, Xisca. Realment una recerca superinteressant i que crec que teniu unes troballes que certament en el final se podran traslladar a la clínica, no? D'alguna manera. Llavors, anem a veure si hi ha alguna pregunta dels assistents. Com que avui ho hem fet d'aquesta manera a través del Zoom, teniu l'opció de fer les preguntes a través del xat, del GoToWebinar, però tal vegada també a través del Zoom, si us resulta més còmode. No sé si algú s'anima. Mira, aquí en el xat tenim en Diego.
Hola, Diego, ¿qué tal?
Bien, bien.
Tengo que poner...
Gracias.
Mira, tu pregunta es cómo de difícil es hacer emirrib a frozen or FFP samples.
Sí, o alguna forma de leer las metilaciones en adenosinas.
Bueno, Claro, el problema del MRIP es que necesitas mucha cantidad. Yo creo que igual esto se puede solucionar con nanopore sequencing. Quizás que además en nanopore, con el MRIP, sabemos más o menos zonas donde está el M6A site, pero no es nucleotide específico, ¿no? Pero con el nanopore, no sé, tendría que hablar con Eva Novoa, pero yo creo que la cantidad de RNA es mucho menor. Sí. No sé si te he contestado.
Una pregunta más, ¿puedo?
Sí.
El nivel de la metilación de las adenosinas en tejido puede ser informativo también.
Sí.
Y para hacer inmunoquímica directamente.
El otro día vi una charla, creo que era en prostate cancer, y miraban diferentes niveles por mass spectrometry, pero de un montón de muestras, y pudieron determinar cómo los diferentes niveles de M6A, pero no con MRIP, con mass spectrometry,
Claro.
para ver a grosso modo, o sea, así no sabrías qué transcriptores están metilados, ¿no? Pero sí que tenían datos muy chulos. Sí.
¿Los anticuerpos van bien?
Sí.
¿Para inmunos? Ok.
Sí.
Perfecto.
Xisca, tens més preguntes en el xat? No sé si les puc llegir.
Mira, sí, les vull llegir. Na Maria Lluïsa Bonat me diu que no té cap preguntes, però sí una gran felicitació. Vull dir que jo vaig descobrir la bioquímica en la Maria Lluïsa Bonat. Va ser la millor professora que he tingut de bioquímica i me va encantar.
Ai, quina emoció, gràcies, Xisca. Una feina estupenda, la teva. I t'he de dir una cosa, una de les diapositives, el resum del teu treball, aquest primer de postdoc, el mostren els alumnes ara. No?
Sí?
Sí. Vinga.
Mira. A veure que jo crec que tenir bons professors així quan començam és important. Encara en la Míriam recordam el primer jornal clar, perquè a més en el laboratori feim jornal clars i tot això, és que el primer jornal clar el vaig fer en la Lluïsa també, i va ser, no sé, va ser molt important per decidir-me. Jo volia fer medicina, a més, no volia fer biologia ni bioquímica ni tenia un laboratori en el nord de Suècia, però sí, va ser un xip, va canviar molt. En Pere Llinàs, moltes gràcies, molt interessant, tenc un dubte. Com heu determinat el citoplasma? És una cohort vostra o ve d'una base de dades? Varen comprar arrays d'aquests que tenen immobilitzat 100 i pico pacients, a Biomax, algo així. O sigui, van comprar uns arrays i van fer immunocytochemistry a TMTT3. Sí. Ara començarem a col·laborar amb Antònia, farem coses xules amb mostres humanes, de veres, això espero. Bueno, no sé qué, gracias por compartir este precioso trabajo, felicidades. ¿La distribución de la marca de metilación de RNA afecta el outcome o el tipo de reader que se une? Sí, he visto dos patrones de unión, a la zona 3' UTR o a todos los exones. Ah, por cierto, soy Miriam. Hola, Miriam. Claro, yo creo que hasta ahora nos hemos centrado mucho en saber o en hacer gene ontology de qué genes están metilados, ¿no? Pero es que da igual. Hay tantos, el consensus es tan inespecífico, o sea, son 4 letras, se puede metilar en tantos lugares que lo importante es saber cómo se reconoce, yo creo. Esto es mi opinión personal. Pero, por ejemplo, sí que la mayoría están en el 3' UTR y la marca en 3' UTR está básicamente relacionada con estabilidad, que lo reconocería el YTACF2 y lo marcaría para degradación. Luego hay otros estudios donde se ha visto que en menos frecuencia hay M6A en el 5' UTR y esto está involucrado o más relacionado con translation. Así que creo que ahora el campo también está muy centralizado en entender qué otras proteínas, como zinc finger protein 217, podrían regular la actividad de la metilasa. Bueno, no sé si te he respondido, pero sí. El outcome no es la metilación, es cómo se lee la metilación. Sí.
Xisca, na Míriam segueix escrivint algunes preguntes més.
Vale, no, no puedes intervenir hablando, vale. Que no, la metilación es cotranscripcional, ¿vale? Por eso también ahora, bueno, hay muchos estudios enseñando que afecta a splicing y tal. Lo que pasa es que la formación del complejo MTTL3, MTTL14 se forma en el citoplasma y luego va al núcleo, ¿vale? Lo de que, a ver, está establecido que la metilación no ocurre en el citoplasma, pero hay unos estudios de virus de estos de mRNA citoplásmico que no van al núcleo y tal, que ven que sí que puede haber en según qué casos metilación en el citoplasma, pero en general no, es cotranscripcional.
Vale, Xisca, de moment no hi ha cap pregunta més tampoc a l'altra banda i jo el que te volia demanar és si finalment aconseguim obtenir mostres de pacients, que crec que vares dir que estaves més interessades en el tipus de triple negatiu, no?
Sí.
I quins experiments o quins assajos faries amb aquestes mostres?
Sí, nosaltres, bueno, com hem fet tots aquests experiments de seqüenciació i veiem que en els knockdowns de MTTL3 hi ha alternative splicing, hi ha patrons d'alternative splicing que són diferents en breast cancer comparat amb normal cells. Val? I hem fet, no he tingut temps de mostrar-ho, però bueno, molt d'aquests transcrits que estan diferencialment, tenen un splicing diferencial, estan involucrats en el fenotip del breast cancer. I nosaltres el que voldríem validar és aquesta dada, ho estem validant actualment amb cèl·lules, però ens agradaria veure si trobam un patró en càncers de pacient.
Vale,
I també una altra cosa que ens agradaria estudiar és com els diferents... Que això serà un projecte de na Marga, que na Marga de divendres és en el meu lava i, pobreta, des que ha arribat està ploguent. O sigui, jo espero que no se me'n vagi pres, que almenys continuï. Però bueno,
aguanta ja.
Que aguanti na Marga. Ja li he fet firmar un contracte d'aguantar-me el temps. No, que també una de les coses que farà na Marga, que té una beca de mobilitat, una PhD Marie Curie, és en el laboratori d'Itàlia, d'Enric Dassi a Trento, mirar com els diferents readers, que està lligat en la pregunta de Míriam, ella el que farà és mirar com els diferents readers s'uneixen en aquests transcrits, no? Amb això, bàsicament, immunoprecipitarà amb els readers i veure amb quins mRNA s'uneixen. I nosaltres també volem veure amb mostres de pacient si aquests readers estan desregulats, o sigui, farien qPCRs, RT-PCR, o sigui, no molt sofisticats. Bàsicament, poder validar que el que estem observant en les cèl·lules també passa en els pacients.
Claro. Bueno, pues crec que ja no tindríem més preguntes. Pues moltíssimes gràcies, ha estat realment superinteressant.
Gràcies.
Que moltes vegades no tenim tantes preguntes, així és que és una molt bona senyal.
Me sap molt de greu els problemes informàtics que han tingut, perquè, bueno, però bé, espero venir qualque dia en persona.
Clar que sí, no te preocupis, esperem que qualque dia puguis venir per aquí, que també sempre és més agradable. Però, bueno, te donem l'enhorabona i esperem seguir en contacte.
Molt bé, gràcies.